So hello friends, welcome back to my channel Medicine 101. Today we'll talk about insulin and the serum potassium levels. So the regulations in between the insulin and the serum potassium level. As we know that the potassium that is one of the most important electrolytes, so it should be maintained, in the narrow range. The potassium level within the serum that should be maintained in the narrow range. Okay, so why it should be maintained? Because if we see that within the serum, if we see the potassium level that is getting increased, that can cause the hyperkalemia. So whenever we get the development of the hyperkalemia, that can cause the cardiac arrest, we can get the cardiac arrhythmia. In the same way, if we get the potassium level, if we get the less amount of the potassium within the circulation or within the serum, that can cause the hypokalemia. So whenever we will get the hypokalemia, that is also responsible for causing the cardiac arrhythmia. Okay, so that's why I should write it over here that potassium level. If we write it over here, the serum potassium level should be maintained in narrow range. Okay, in narrow or range. Now try to understand. If you just draw the graph for it, just to show the value for it. If I say, for example, the potassium level over here, I'm just taking the potassium level 3.5 milliequivalent or the millimole. We can write in terms of the milliequivalent or the millimole per liter. So I'm writing milliequivalent per liter. Okay, is the lower border for the potassium level, and the upper border is 5.5. So if you write 5.5 milliequivalent per liter. Okay, in this cases, in between that, the serum potassium level that should be there. So the serum potassium level that should lie in between or 3.5 milliosmol, or we can say the milliequivalent or the millimole per liter. We can say, or the upper limit there is 5.5 millimole or milliequivalent per liter. Okay, so in between these things, the range should lie over here. Okay, now if you see more than that level, more than the upper limit, that will cause the hyperkalemia. So whenever we will get the hyperkalemia, at the time we'll see the problem where the heart, so that can cause the cardiac arrhythmia. In fact, it can cause also the cardiac arrest. So it can cause the cardiac arrest. In the same way, whenever we will get the potassium level that will be less than the 3.5 milliequivalent per liter, this thing is known as hypokalemia. At the time we will also see the cardiac arrhythmia. Now, how can you control the potassium level? That's the main thing. And what's the relationship in controlling the serum potassium level with insulin? That's the today's main topic. Okay, so for that thing, let's talk some very basic points, then I'll go in details. So okay, see, every day try to understand. Every day we take near about 100 to 200. Try to understand. If I say every day we take 100 to 200 milliequivalent, or you can say the millimole per liter of the potassium. Try to understand. Before that, let's have some idea the potassium level within the cell and outside the cell. If I say this is the normal circulations, the general circulations. Okay, and for example, I'm taking is the normal cells. I'm taking the cells over here. Okay, and for example, this is GIT. I'm showing that. So try to understand what do you know. Normally, the value for the potassium within the outside the outside the cell, that means in the extracellular compartment, that will be how much? In between 5.5 to 3.5 millimole per liter. And what I would the cells. So if you write it over here, we'll get around, for example, I'm just taking the average value. For example, I'm taking 4.5 milliequivalent per liter. I'm just taking in these values. Okay. And if we say in terms of the body fluid distributions, so what do you know? If I say, for example, there is a young adult man who is having the 70 kilo weight, so 60% that will be the total body weight. So out of the 60%, that means if we get, try to understand, if we get the total body weight 70 kg, so we'll get 60%, that will be total body weight. So 60%, that means we'll get around 42 or 40. Okay, so let's write it over here. We'll get around how much? So we get here 60%. That will be the what? That will be total body weight. Total body weight. Okay, so that will be the how much? 60% of the total body weight. That means the total weight. How much is 70 kg? That will be the total body water over here. It will be the total body water over here. Okay. Now see, if I just say from this example, so we'll get around, for example, 42 liter we are getting, 42 liter. Out of the 42 liter, I have already explained in the separate video, in the edema part, you will see over there that we'll get 2/3 where within the intercellular fluid and 1/3 will get extracellular. Within the extracellular, the blood will be also included over here. Okay, so if we say we'll get around how much? We'll get around 28 liter where within the cell, and we'll get 14 liter where within the extracellular. That means in the circulations also. Okay. Now let's count the normal. Let's do the normal mathematics over here. So what do we know? That the potassium level where within the cell is how much? Around 140 milliequivalent per liter, 130 milliequivalent per liter. Okay. Now if you get it over here in terms of the total amount, how much? 28 liter in the whole body. In the total body, if we say what will be the total potassium level within the cell? So in the whole body. So if you write it about the potassium, total potassium, will get how much? It should be 140 multiply 28, this much milli. Okay, let's write it over here. Milli equivalent per liter. In 1 liter, we will get how much? 140, then 28, how much? This is the normal mathematics. We have to do the product over here. So we will get around how much? We will get around 4000. Let's write it over here. 4000 milli equivalent per liter. Okay. And if you just do the normal, mathematics over here, how much? Is the 14 liter, we will get the how much? The extracellular fluid, 14 liter, and the potassium level in the extracellular, how much? 4.5 milli equivalent. So we get around 60. We get around 60. So 60 milli equivalent per liter we will get over here. Okay. This is a normal thing. Now see, this value should be controlled always. This value should be controlled always. That value should be maintained everywhere. Now what is the problem? For example, whenever you are taking, basically whenever through the diet, okay, whenever you are just eating the food, at that time, whenever the glucose, they will come over here. This glucose along with the glucose will get the different kinds of the other nutrients. Okay, the same way from here, these things will be absorbed. From this cells, these things will be absorbed. Along with that, we'll get also the potassium. So every day, if we say, whenever through the dietary systems, so through the diet, especially if we just take the fruits and vegetables, the fruits and vegetables, they are rich in potassium. Okay, they are rich in potassium. So whenever we will get the more amount of the potassium, so more amount of the what? Potassium, that will come over here, more amount of the potassium. So in the same way, like glucose and other nutrients that freely absorbed from here, in the same way, this potassium, they will be not checked properly. The potassium will be not checked properly over here. They will be also absorbed freely. So whenever the potassium, they are also absorbed in the same way. For example, the glucose is coming over here, we will get the glucose, we will get the other nutrients. In the same way, we will get also the what? This potassium, they will be also freely absorbed. So whenever this potassium, they will be freely absorbed over here, they will be freely absorbed. Now try to understand, every day we take near about 100 to 200 milli equivalent per liter. This much potassium we take every day through the fruits or the vegetables, you can say. Now whenever this much amount of the potassium that will be absorbed over, definitely try to understand within the circulation, so the potassium level, that will be much more, and that can cause what? Hyperkalemia. Whenever we will get the hyperkalemia, that will cause the cardiac arrest. Now definitely within the body, because when every day we are taking the potassiums, but we are not facing this type of the problem. But try to understand, every day 2 to 3 times we will get, whenever we are just eating the food at the time, the potassium level, that will be elevated, and that will threaten the heart over here, and that can just, there is chances for the development of the cardiac arrest. But under normal circumstances, if we see under normal circumstances in the physiological conditions, we don't face this type of the conditions. We don't face this type of the problem. Why? Because definitely within the body there are some, the counter regulatory mechanisms or the alternative mechanisms that will handle these things. So what is that thing? Try to understand from here. What do we know? Through the nephrons, through the kidneys, draw it more properly. So what do we know? Through the kidney, these potassiums that will be expelled out. So the potassium through the kidney, through the lymph systems, what? The potassium that will be expelled out. That will be expelled out. Now try to understand, if we say over here that in these portions, what do we know? The potassium that will be reabsorbed. Potassium that will be reabsorbed. In these portions also what? Potassium that will be reabsorbed. And in these portions from the blood, the potassium that will be secreted. From the circulations, the potassium that will be secreted. So this potassium that will come out through the urine outside. So you can say 98%, or we can say, we can say 90% of the potassiums, whatever it is coming out, the 90% of the potassium that should be excreted out through the urine over here. And 10%, the 10% that will come through the fecal matter. Fecal matter. Now try to understand, this is the balance. Whatever the potassiums, for example, here it was 4.5. Now it should be always 4.5. So for example, if we take 100 milliequivalent per liter, if we say 100 milliequivalent of the potassium we have taken, so the 100 milliequivalents of the potassium should be excreted out. If I take 200 milliequivalent potassium, then 200 milliequivalent of the potassium should be excreted out by the urinary systems over here. Okay, but what are the problem we'll see over here, that whenever the potassiums are increasing in the the speed of getting increased within the circulations for the potassium level, in the same speed the kidney is not able to excrete it out. So try to understand what I'm saying. I'm repeating once again. For example, the potassiums, the accelerations of the potassiums, the accelerated speed we can say over here, or we can say that the concentrations of the potassium within the circulations after taking the diet, that will be increased so rapidly that the kidney, they are not able to maintain that much speed to excrete out. As a result, we see there is the chances for the accumulations of the potassium within the circulations that can cause the partial, or you can say the what? Temporary hyperkalemia, because the cardiac arrest. So definitely there should be some mechanisms that the potassiums from the circulation should be shifted in other portions. Okay, and whenever the potassium that will be back to the normal level over here, then the cells excreted out. The cells, by the process of what? Exocytosis process, the potassium should be come out, and from there the potassium that reach where? Within the nephrons, and from the nephron it should come out. Okay, so whatever the mechanism is over there, that's I'm going to talk now. Okay, so what type of mechanisms we'll get. For that thing I need to draw, I need to erase these portions. I think they need to be better for you guys. Okay, so hope it's clear up to this much. If you have any questions, you can let me know in the comment section. Okay, now what do we know, that whenever you're just taking the food, try to understand. Okay, I'm not erasing this part. So whenever you're taking the food, for example, you are taking the food, at that time, try to understand, the glucose is coming within the GIT, so glucose that will be absorbed from the GIT, and from here what do we know, that it will come within the general circulations. Whenever it will come to the general circulations, then from the general circulations it will be taken by the cells. And in these cases, what do you know, that whenever the amount of the glucose within the blood that will be elevated, at the time the insulin that will be activated. And what the insulin they will do, see, from the pancreatic beta cells, what do you know, from the from the pancreatic beta cells, what do you know, from here the insulin that will be excreted, so insulin that will be produced. Anyway, so whenever we will get the insulin that will be produced from here, so insulin will come over here, and what the insulin will do, this insulin will push the glucose molecules. This insulin that will push the glucose molecule where? Within the cell. So this glucose molecule, this insulin that will push the glucose molecule through the blood receptors where? Within the cells. In the same way, this insulin they will also be able to push the amino acid where? Within the cell. So they will also push the amino acids within the cell. Amino acids. In the same way, we will also see this insulin, they are having also the capacity to push the potassium where? Within the cell. How they are doing this? That's the main topic for today. So they will be also able to because whenever you are taking the diets, definitely the potassium that will be also reached where within the general circulations, and from the general circulations the insulin is also helping the potassium to go where within the cell. Now how it's helping, that's the main topic for today. Okay, now listen to me very carefully how I'm explaining these things. Okay, now let's continue. I'm erasing this part, guys, because in this time we need to use these portions, whole portions. Okay, now try to understand. Okay, yes. So is this GIT? Okay, let's try to mode. Proper, yes. Now we have taken the food. Along with the food, what do we know? The glucose that has come out where within the GIT, and from here through the absorption method, what do we know? That these things that will be taken where within the general circulation. So the glucose molecules of the nutrients, not only glucose, the amino acids, the fats, all these things that will be taken where within the circulations, and from here we will know what these things ultimately that will reach where within the cells. 3.5. Okay, and we will get the different kinds of the transporters. So glucose, we will get the amino acids, we will get also the potassium. That is the most important thing. Now how insulin is helping in the potassium, to push the potassium within the cells. That's today's main topic. For that thing, I'll just draw one cell, hypothetical cell, for example the muscular cells where the the insulin receptors are present. So for example, I'm just taking this the cell where the insulin receptor is present. So definitely see, whenever the glucose level is getting increased, so whenever the glucose level that is getting increased, definitely the insulin level that will be also increase. So whenever the insulin is getting increased, how do they work? I have already explained how the insulin or the transmembrane pathway or the mechanisms by which the insulin will go where within the cells, and whatever the functions they will do attaching with the receptors. So for that thing, let's draw. There is the presence of the insulin receptors. Within the insulin receptors, what do you know? There is the 2 alpha units, and we get the 2. Okay, let's draw with a different color. We get the 2 beta units. So beta units, I'm repeating once again, we'll get the beta units. Now try to understand from here. See, whenever the insulin that will come over here and that will get attached to these regions, we'll get some conformational changes within the alpha parts, and there is the disulfide bonding in between them over here to here. Okay, and whenever we get the transformation changes within the alpha part, these things that the information that will be transformed where within the beta units. So we know within the beta units there is the presence of one enzyme that is known as tyrosine kinase, and within the beta units, as it is the tetrameric put in the insulin, so definitely there will be the presence of amino acids, where we will get the presence of the tyrosine amino acids, where we will get the presence of tyrosine amino acid. Now what it will do, this tyrosine kinase enzyme that will just do the cross autophosphorylations. That means whenever this tyrosine kinase enzyme that will be activated, that will do the phosphorylations of the opposite part over here, opposite part. And this, whenever it will be activated, it will just do the phosphorylation of this part. Now this beta units, they will be excited, they will start dancing, for example. So they will wait for another molecules to get stacked over here, and we know there is a presence of one unit over here within the cell that is known as insulin receptor substrate, where within the cytoplasm within the cell. So whenever this beta units they will be activated, they will be the site to get attached for insulin receptor substrate. So insulin receptor substrate that will come over here, and whenever it will come over here, at that time whatever the changes we will see, there is also presence of the what? Tyrosine amino acids. There is also presence of the tyrosine amino acids. 20 to 22 amino acids will get over here. What it will do, this tyrosine kinase, this enzyme, this will also do the phosphorylation of this part. So whenever it will just do also the phosphorylation of what? The tyrosine of what? The insulin receptor substrate. So it will also do the phosphorylations over here, and whenever it will be phosphorylated, try to understand. this insulin receptor substrate that will be activated, and it will just open certain domain. So whenever it will be activated, it will open certain kinds of the domain for some molecules. Here will also get opening of certain types of the domains. Now this is known as SAH2. Now what is SAH2? It is the what? Circ homology. Circ homology. Okay. Now here we get binding of on enzymes. So you can say the protein, whether we can say the protein or enzymes. So whenever this protein that will be active, active over here. And what is the function of this protein? I will tell the name of this protein later, but first let's know the function of this protein. What this protein will do? What do you know normally is the phospholipid layer, or is the membrane. What do you know in the membrane? There is the phospholipid bilayer. Okay. So within the phospholipid bilayer, what do you know? There is the presence of presence of what? I try to understand from here. Phospholipid bilayer over here. If we see the structures over here, for example, here we will get the presence of one compound that is known as phosphatidyl inositol diphosphate. Phosphatidyl inositol diphosphate. Okay, is the phosphatidyl, it is inositol group, and here we will get in the number 4th and number 5th positions, we will get the phosphate molecule. We will get the phosphate molecules. That side is known as PIP2. PIP2. Now, what is the functions of this protein of the enzyme? Okay, this enzyme, they will add one more phosphate. This will add one more phosphate over in the 3rd positions. In the 3rd positions, it will just add one more phosphate. Now, whenever this will add one more phosphate over in the 3rd positions of the inositol compound, now it will be converted into what? PIP2 to PI3P. That means now we can call it as what? Phosphatidyl inositol 3-phosphate. Now, whenever we get this type of thing, see these things, these molecules, that means phosphatidyl inositol 3-phosphate activated, and whenever it will be activated, try to let's show these portions over here. Okay, now see there is the phosphate molecule, there is the phosphate molecule, and there is a new phosphate molecule that has been added by, what's the name of this enzyme? It is adding the phosphate at the number 3 positions of the inositol compound. That's why it should be called as what? Phosphatidyl inositol 3 kinase. Phosphatidyl inositol 3 kinase. So let's write it over here. The name of this enzyme, the same enzyme will get over here. So we'll get phosphatidyl inositol 3 kinase, or you can say PI3K. Okay. Now, whenever it will be converted into from PIP2 to PI3P, at that time, these things, you will have certain molecule. This molecule will be activated, and it will have certain pockets that will be activated. And whenever these pockets will be activated, whatever the thing will get over here, this will actually have certain types of pockets where certain types of the molecule they will get bind. Now, what are the names of these molecules? The first, let's talk about these ones. The name of this molecule that is known as PKB, or the protein kinase B, or you can say the AKT. Okay, and what this molecule will do? This molecule will add one more phosphate over here. So one more phosphate over here within the protein kinase B. On the protein kinase B, one more phosphate will be added by the actions of another molecule that is present over here. So let's add the phosphate first. Then I'm saying what is the name of this one. So the name for this one that is known as phosphatidyl inositol dependent kinase 1. So the name for this one enzyme that is known as phosphatidyl, okay, inositol dependent kinase 1. Phosphatidyl inositol dependent kinase 1. Okay. Now it will just do one more. It will add one more phosphate in these positions in the protein kinase B. Whenever one more phosphate that will be added over here, this molecules will be activated, and whenever it will be activated, it will do? It will just give multiple signals within the cells. Multiple signals within the cells. One signals that will just for example, one signal will come that will uptake the glucose molecules within the cell. It will activate the different kinds of the GLUT receptors over here. Okay, and one more signal will come over here that I will talk now. Okay, that will activate one more enzyme that is known as protein phosphatase 1. That is known as protein phosphatase 1. Now it is the protein phosphatase 1. Now we'll talk about another part of the story, then I will just complete it. Okay. Now, so what do you know within the cells? There is a presence of the what? Sodium potassium ATPase pump, right? Sodium potassium ATPase pump. So what do we know in terms of the sodium potassium ATPase pump? By the actions of the what? By the action of ATP, whenever ATP will get breakdown into ADP plus phosphate, at that time the ATP will be utilized and it will do against the concentration gradient. It will just throw out the 3 molecules of the sodium outside and it will take 2 molecules of the potassium inside. Now within this portion, within this pump, we'll get certain, if we can just do the phosphorylations in some portions, if we can add the if we can add the phosphate group over here, this will be stimulated. This will be stimulated by what? By using the ATP or doing these things. But there is a presence of another kind of the phosphate. For example, there is the presence of another kind of the phosphate. This is known as the inhibitor. This phosphate is responsible for inhibition. They will do the inhibition functions. This kind of the phosphate. Now, what the protein phosphatase 1, that will be activated by what? That will be activated by the protein kinase B. The protein kinase B that will activate the protein phosphatase 1. This protein phosphatase 1, what they will do? This protein phosphatase 1, try to understand. They will go where? They will go within the sodium potassium ATPase pump and they will just block the actions of the they will just remove this phosphate group. I am repeating once again. See, the protein kinase B or the AKT molecules, whenever they will be activated, they will give many signals. One of the signals that will activate the protein phosphatase 1 enzymes. So whenever the protein phosphatase, from the term phosphatase means what? That will remove the phosphate group. So whenever the protein phosphatase 1, this will be activated, this enzyme, this will remove the inhibitory phosphate group from where? From the sodium potassium ATPase pump. So whenever this phosphate group that will be removed from here, definitely more amount of the what we will see, and the more amount of the sodium potassium ATPase pump that will be activated more, and more amount of the what? The sodium that will be thrown out, and the more amount of the potassium, that will be taken within the cells. Like this, we will get multiple receptor, multiple molecules over here in terms of the sodium potassium ATPase. Okay, here we also get the sodium that will be thrown out, and we'll get the 2 molecules of the potassium that will be taken in. The same way, 3 molecules of the sodium, the 2 molecules of the potassium taken in, and the 3 molecules of the sodium that will be thrown outside of the cells. Like this, we'll get the mechanisms how the potassium, okay, whatever the extracellular potassium that was over here, okay, that will be taken within the cells by the actions of the insulin, and what's the intercellular mechanisms I have already explained over here. So if you think this video is helpful for you, please do like and subscribe my channel, and see you in the next video soon. Okay, thanks for watching.